Okay.
And remember to say next week we start the same time. Redovan said he can start 1 hour earlier. So then we don't have to switch twice for the fun.
But next week, you have not changed time yet.
We only changed in 2 weeks.
In 2 weeks. Yeah, so next week is at
we start
4.
the same time. And it's on YouTube. We are live.
Okay, perfect. Good. Okay, so I think we can get started. So good morning and afternoon, everyone. Thanks for being here and joining us today for our second seminar of the fine season. My name is Adriana Maldonado-Chaparro. I'm a professor at Universidad del Rosario in Colombia, and I'm hosting today's seminar. Well, before getting started, I want to take some minutes to make some announcements. First, I would like to thank our previous speaker, Dr. Alex Ophir from Cornell University, for a fantastic opening talk and discussion on variation in parental care and the impacts on social brain, social behavior, and reproductive decision-making. Second, I would like to announce our speaker for next week, Dr. Redon Barry. I hope I pronounced that correctly. It's a little bit hard for me. And he'll be talking about putting biology into cooperation theory. We look forward to his talk. Just remember, everyone, the US changed time, and so it will be in Europe 4 in the afternoon, not 5 as usual. So just keep that in mind. Also, a few reminders about the fine structure. Just everyone is on the same page. This is a 50-minute seminar about each, 45 to 50. It's going to be followed by a maximum 60 minutes of discussion. During the seminar, please make sure to keep your microphones muted. Only turn it on when we get into the discussion after the talk. And then during the discussion, please type the question mark in the chat. I will send a message to everyone just to remind. And if you are watching the stream on YouTube, please type in your questions. The moderator will relay them to me, and we're gonna relay them to the speaker. So we encourage everyone on YouTube to type in questions and engage live in this seminar. Well, with no further ado, I'm going to introduce our speaker for this week, Dr. Floria Mora-Kepfer, UI. Floria got her PhD at the University of Miami in 2011. She's currently an assistant professor at University of Rochester, and it's the academic director of the Galapagos Study Abroad program. So if anyone wants to know a little bit more about Galapagos, please get in touch. She leads the TROP Biolab, from where she tries to build a more diverse and welcoming environment for students in STEM. Her research focuses on understanding the evolution of cooperation and the selective pressures that favor cooperative group living, particularly the relationships between the brain development and sociality in animals. She's broadly interested in understanding how do animals process information from their physical and social environment to make decisions that enhance their survival and reproductive success. More recently, she's exploring the effect of behavioral manipulation by parasites, something that she will be talking about today, in insect societies. And to answer these questions, studies, well, insects that vary in the extent of sociality. So we'll be hearing about these flexible social systems in wasps. Well, in addition to her awesome science, Floria is also very excited about being a Latina scientist, and she's highly committed to diversity. So get in touch if you want to learn more about how to be a Latina scientist. And with this, our seminar title is Mechanisms Underlying Parasitic Manipulation of Social Wasps. Floria, thank you very much for accepting our invitation, and the mic is all yours.
Thank you so much for the wonderful invitation. Thank you to Adriana, my host, and to the whole fine organizers for giving me the opportunity to share some of our newest work. So I think I can share my screen right now, correct? Let's see. Can everybody see this well? Wonderful. Okay.
But it's
so
not fair. We see your, okay, now it is.
Can you see it now? Yeah, it takes a little bit, I think, of time to kick in. Good. Okay. So again, thank you so much for the invitation. I'm very excited to talk about some of the work that we started just as I moved to the University of Rochester and during the pandemic. So I am really grateful for the wonderful team of students that I have and trainees that have been just really troopers throughout the challenges that we've had. And I'm going to be focusing on mechanisms underlying parasitic manipulation of flexible societies of wasps. So let me see if I can move forward. Yes. So when we think about parasitism, it really is a very successful and widespread strategy across many different taxa. It really has there are many instances in different systems across animals where we see wonderful studies of coevolution in hosts and parasites with diversity of systems, and also the arms race that is unique between hosts and parasites. But what we at this point know less about is what are the mechanisms that these parasites use to manipulate their hosts. And that is what our group is very interested in starting to elucidate. So the big question is that parasites could manipulate their hosts in several ways. One is to interfere with this communication, the immune and neurocommunication that the host has. They could also secrete substances to alter neuronal activity, and they could also, or they could induce proteomic-based or genomic-based changes in their host to signal different pathways and use the plasticity, the phenotypic plasticity of their host potentially to manipulate the host to secure fitness and survival of the parasite. And on top of this, what's really exciting for me as someone that studies social evolution is that social hosts are what I call ideal vessels for infection, because not only individuals can get infected per se, but if manipulated the right way, they can give open the door for the parasites to be able to infect more individuals within the same group. So we've seen very elegant work of other scientists that are studying the mechanisms underlying parasitic manipulation in social insects, such as Suzanne Foytzik with this tapeworm that manipulates ants, and João Araujo and other collaborators that work on Ophiocordyceps and how it manipulates their ant host. So it seems to be that phenotypic plasticity is a big player in the manipulation of social hosts. And something that hasn't been explored as much is there really a cost to phenotypic plasticity? And in the past, I've been lucky enough to collaborate with Dr. Laura Beani, which you see in this image. And she, in turn, had strong collaborations with Bill Hamilton. And they had this very interesting exchange early on, I think this was in the 90s, where they talked about how parasitic manipulation can really take over the plasticity of the host. And this would be particularly relevant in species that had morphological, behavioral, and or physiological activity. And Hamilton was particularly keen on extranidal aggregations. So there were these social wasps that were infected by these parasites, who are also insects, these parasitic insects of the order Strepsiptera. So strepsipterans are not a very well-known order. It's a small order of parasitic insects. And in their collaboration, when they talked about, after all, if they can so completely castrate the wasp without killing them, an ability obviously evolved in an association through an immense period of time. It will not be surprising to find that they also manipulate the behavior. And this collaboration really catapulted Laura's work, who she has also been a mentor to me. And now I'm very lucky to continue along this line of work to try to understand what are these costs of phenotypic plasticity in these social hosts. Recently, in collaboration, when I was at Cornell University with Mike Sheehan's lab, we had some work in which we wanted to test for mechanisms underlying caste determination in behavioral responses and dominance hierarchies. So there's a wealth of information in social insects and from previous research that social wasps, the ones we study, have flexible dominance hierarchies. So potentially anybody could be a queen, and reproductive and social dominance is established by a combination of social interactions, so aggressive interactions, make an individual be the top rank in a hierarchy, and also hormonal inhibition of ovaries of the other females in the colony. But if the queen dies or is replaced, taken over, somebody else can develop their ovaries quickly. So this means that even within adults, there's strong plasticity, physiological and behavioral in this system. So we went and we wanted to see if there were neurogenomic responses. That is, if we put individuals in diets where they had to compete for social dominance, could we see physiological and neurogenomic responses? And what you see here is a figure where the winners of the contest are in red and the losers are in blue. And you see several genes here. And what we see is that the losers had strong changes in gene expression after losing their battle. So this was very exciting to us, this confirmation where there seems to be strong neurogenomic flexibility and plasticity associated to responding to social interactions as adults. So this was confirmation that there is also neurogenomic strong plasticity in the system. So with that, I wanted to introduce a little bit of our model system, the Polistes social wasps. In temperate zones, wasps initiate nests in the spring. In the early summer, the first brood emerges adults, and these are very important because they're the first set of subordinate workers, which will increase productivity in the colony. And by the late summer, after several generations of workers, the colony will, the foundress will produce giants, and I'm going to refer it as giants as future queens, and males. They mate. Everybody in the colony dies, except these giants, which will be the future queens the following spring, which overwinter. And then the cycle begins again with these giants becoming foundresses and starting out the colony cycle again. And then they have to figure out who will be the dominant individual, and this will be via the behavioral aggression and the dominant and the inhibition of the ovaries. Now, the plot twist to the story is that these Xenos parasites, so these strepsipteran parasites, can infect, but due to the life cycle of the parasite, they infect only workers usually, and these workers are produced early in the season. So they don't have the chance to, in some cases, there might be some overlap with future queens, but the really prime moment of the season is when the nests are being established. So these develop inside the abdominal cavity of the host. I hope that you can see my cursor, but you see a female parasite with their cephalothorax sticking out of the host. And this hole you see in the abdomen is where a male parasite emerged. So they are very sexually dimorphic. The interesting thing is that these workers that are infected, they will never perform any tasks for the colony, and eventually they will abandon the colony. So they are supposed to be workers and indeed work for the colony, but the parasite is manipulating them to not do any of their usual social tasks. And what is even more compelling is that these workers are supposed to be short-lived and they should just die at the end of the fall, and instead their lifespan gets extended by the host and they overwinter like these giants, aka the future queens. And we really wanted to figure out how these parasites are taking over the phenotypic plasticity of their social hosts to do this. So what I'm going to be talking about today is this relationship between why and how questions of social hosts being manipulated by their parasites, where we wanted to understand first cognition and behavior, how the infection cycle happens, and when individuals are infected, how this will influence changes at gene expression and neural levels, which will indeed also trigger physiological and behavioral changes of these workers, which should enhance transmission of new parasites and the infection cycle to happen again. So the first question I wanted to answer, when our group wanted to answer, is how do parasites really recognize their hosts? The second one is, can parasites infect novel host species? Third, what are the molecular underpinnings? So what are these mechanisms that are being used by parasites to manipulate their hosts? And finally, if we could start to get actual gene networks that could be manipulated by the parasite, indeed. So to start with the first question, because this is a poorly studied system, at least in North America, we wanted to, I want to briefly introduce the life cycle. So early in the spring, a wasp or a small group of wasps will initiate a nest. If these are parasite larvae, so if they make it to the nest, and we'll talk about this in a second, they are able to infect the host. So our jelly bean looking structure here is a wasp host larva, and the little larva inside is the parasite, so it's able to successfully infect. Then the adults, the wasp metamorphosize into the adult, and you could see here in gray where the parasites are lodged inside the abdominal cavity, and the parasitic manipulation should be happening at this point. And then we have female and male parasites. The females remain as obligate endoparasites, so they will always be inside their wasp host. And the males instead, they emerge as free-flying adults. And which again, this is a giant one, it's not up to scale, but this is what a male looks like. And then the male will locate a female and inseminate via traumatic insemination in the cephalothorax. So it will inject sperm into the cephalothorax of the female parasite. And the female parasite becomes gravid, but it remains inside its wasp host. So a pretty complex and pretty unique system at the time, showing some of the most sexually dimorphic insects that you can think of. So our first hypothesis was that perhaps this gravid parasite female, if we focus on after what happens in this figure G, so a gravid parasite should manipulate the host sensory systems to find the right host nest to be able to release their larvae, so the larvae can infect the wasp host larvae. And a second hypothesis, which is not necessarily mutually exclusive, is that the parasite larvae should also be able to recognize and choose their host larvae. So, and at the same time, we're very lucky that we have ideal parallel systems. In North America, we have Polistes fuscatus, which is the wasp here in the bottom, the brown one, which has its own species of strepsiptera called Xenos peckii. And in Europe, we have Polistes dominula, which has been studied by Laura Beani, Fabio Manfredini, and other wonderful collaborators, that they have their own species of parasite, Xenos visperum. So this allows us to work with ideal parallel systems in Europe and in North America. And to top this off, Polistes dominula, our yellow wasp on the top, is invasive in the United States too. And in a bunch of other places in the world, actually. It's pretty cosmopolitan right now. So to be able to answer these questions about the first step of manipulating the host sensory systems, which should be used for social interactions and instead would be manipulated to benefit the parasite, we first go and collect field wasps. We find wasps that are infected with gravid parasites and uninfected wasps. We establish colonies of an uninfected wasps in the greenhouse so we can experimentally infect them. And we rear both the invasive Polistes dominula and the native species. And this work has been really a product of Team Wasps, undergraduate Team Wasps, wonderful work. Apologies, I live close to a hospital, so you might hear some sirens. So for the first hypothesis, to test if gravid parasites hijacks the sensory systems of the host, Federico Sanchez Vargas, which is a wonderful undergrad, helped with another undergrad in the lab, Joe. They set out a very clever experiment in which they had a worker wasp with a female parasite here on the bottom. They're inside a cage, and they placed a nest of Polistes dominula, which is the yellow invasive wasp, and on the other side, a nest of the native wasp. And since we are testing for the effect of a native North American host and parasite, we would expect that the gravid female should be able to manipulate its host to pick a nest of its own species, which should be the brown here to the right, which is the native one. Again, these were unattended. There were no adults. They just had these beautiful larvae ready to be infected by the parasite. So when Faye and Joe ran this experiment, we got really neat results in which it seems that they really, the parasite seems to be manipulating the wasp host to go into its own species and deposit the larvae. We checked for the nest later, and we could see the parasite larvae had been released into the native host species, which was we expected, and we didn't find, and there was less time spent on the invasive host, which would be the wrong host, and we didn't find any parasite larvae after these interactions. So this was our first confirmation that there seems to be choosing the conspecifics nest and releasing larvae on the right host. So the second part we wanted to talk about was how the parasite larvae can, once they're being dropped off, can they really recognize and choose their host larvae? And as Laura Beani very beautifully put it a while ago, triangulans are these parasitic larvae, and she has this hypothesis that triangulans are stupid, that they don't know how to choose their host. So we set out to do this. We put an invasive host that shouldn't be chosen by the parasite larvae. The parasite larvae are these little dots that you see over here, and we put another native wasp. So we have a little bit of a wasp larva jail situation here where they can't move out. And we saw if the parasites chose the invasive or the native wasp. So again, these are the larvae that would be infecting the host. And what we found is that many of these larvae don't make a choice. And when they do make a choice, it's very similar between the invasive and the native. So it seems that the parasite doesn't prefer the native host. Now, the next question here was, well, if they are able to, if they are exposed to novel hosts, can they infect them? And one of the reasons why we were interested in this topic is because it seems that the original hosts of Xenidae, which is this family of strepsipteran parasites, were most likely social wasps. And overall, they're specialists, but they can switch to ecologically similar hosts. So it seems that they have a preferred host, but they can infect more than one species of wasp. And given that Polistes dominula has become prevalent in the United States, we wanted to test if ecologically they would be considered similar hosts. So Maggie, which is another wonderful undergrad student, she's now in grad school, set out to test this experiment. So here we could see our wasp abdomen. We see our gravid female parasite here. We can stimulate it to release. So we, in a way, we kind of milk them for parasites. And here in the bottom picture, you could see two of these parasite larvae that are they are on top of a host larvae, and they're starting to burrow in and infect. we're able to experimentally infect individuals, so expose them to larvae, incubate them, and then check if they're developing inside the host. In this picture, you could see a wonderful head capsule of a host larvae, and these little dots on the side are the parasites. So what Maggie found here is that they were able to enter the host similarly, both invasive and native hosts, and they were able to start developing inside the invasive host because they were even able to molt and progress to the next developmental instar of the parasite. And we were sort of surprised because they were doing similarly in invasive and native hosts. So in a nutshell, it seems that this is an invasive host species, the parasites are capable of infecting this invasive host, but it seems to be, so far, we've only been able to experimentally infect in male hosts. Keep this in mind because this will be important for the mechanism and neurogenomic parts of the talk. So it seems that if you're an invasive host, the parasites have a better chance of infecting the males. And we lovingly call this our Frankenstein wasp because this was an experimental infection of the wrong host species. And you can actually see the parasites over here. So this is just a short video to show you that although they can infect and start to develop inside the invasive host, here we could see the large parasites sticking out of the host. This poor male host wasp isn't doing too well. And we suspect that this has to do with the immune system and how males could be secondary hosts. So the last thing I wanted to talk about to move on to the mechanistic part of the talk is that we also know that individuals that are not exposed to their usual parasites or natural enemies, they tend to stop evolving defenses against parasites and natural enemies that they don't have a need for anymore. So we wanted to see if the parasites could be detected by the social wasp species, as taking care of the offspring is such an important thing in a social insect colony. And if they perform hygienic behavior, if they could perceive that individuals were infected, some of the brood were infected. So here, Maggie experimentally infected wasps in the same setup, checked for hygienic behavior at the day after they were infected for 7 days straight, and on the last day checked for the removal of the infected larvae. And this was done for the invasive host and for the native host, which should be the host of choice, of course. And what Maggie found is that the probability of being removed ends up being pretty much the same by day 7, but it seems that the invasive adults can detect that larvae are infected slightly earlier than the native wasps. So this was pretty interesting to us. And it allows us to start to get a little bit more at the arms race between these hosts and parasites. But we got pretty similar results where infected individuals can be removed if detected. And the other thing is the individual immune response of the host per se. So can they perceive that they have been infected and use their immune system to encapsulate and get rid of their parasite? And we also looked at rates of encapsulation, and we also have data for its effect on ovaries because hosts get castrated when they are infected by parasites. We don't have that data yet, but what we do know is that invasive and native hosts tend to have similar rates of encapsulation. So it seems like the arms race is still happening, and it might not be so different if it is an invasive versus a native host. So that being said, we wanted to move on now that we know that parasites can recognize their hosts and they can manipulate their usual sensory systems to find other nests of conspecifics to infect, and that parasites can also infect novel hosts. We wanted to really look at the molecular mechanisms of parasitic manipulation. So what do we know? What about this system at all? Well, our good colleague and collaborator, Amy Toth, and also Federico Cappa and Laura Beani in Europe, they had worked in the Polistes dominula, so the invasive yellow wasp in its native European range when it's exposed to its native parasite. And Amy had a great paper, which gave us a lot of the background for what we're working on, where they tested for workers that are not infected. Here in the first graph, they compared their transcriptomic brain profiles to infected workers and to giants, which are these future queens. And what we see here is that the infected wasps seem to be an in-between workers and future queens. And there seems to be a manipulation by the parasite in gene expression. Now, the interesting thing here is that male wasps do not show changes in behavior. They still try to mate with females. In social insects, males are not part of the social group, but they do mate with females. And the males do not lose their mating behavior. They're not castrated. So they still produce sperm, which we see here on the second figure, where they confirmed that the male reproductive organs of infected and uninfected males were similar, and they were producing sperm. And we also didn't know gene expression. So we find that this parasite has very strong effects in its social female hosts. It castrates them. It extends the lifespan of workers and makes them have this abnormal behavior when they're not social anymore. But it doesn't have those physiological or behavioral effects in male hosts. And nothing had been known at the time about gene expression, any type of manipulation signatures inside male hosts. So former master's student in my lab, Christine, she set out with a clever neuroanatomy experiment where we had predicted that perhaps, given that infected wasps are manipulated to not be social, to not form any task colonies, this would influence the allocation of energy towards brain structures that are used for the very crucial social interactions, visual and olfactory sensory input. And what Christine found was pretty interesting, because if we compare on the first figure, we have female hosts. We compared for the neuroanatomy, so brain regions, central brain compared to the sensory brain regions, that being visual and olfactory. And if you see, there's no difference between infected females, which are in blue, uninfected workers, so infected workers, uninfected workers, and giants, so these future queens. But when we compare infected male hosts, which is the second figure, we see a strong allometric change in infected hosts, with our here in purple, compared to male hosts that are not infected. So it seems to manipulate differential investment in brain regions in male hosts, but not in the female hosts. So this allowed us to think, well, perhaps if neuroanatomy isn't being manipulated in female hosts, this has to be more along the lines of perhaps gene expression, proteomic changes, neuromodulation, but not necessarily changes in the actual structures per se. The other interesting thing that Christine found is that she compared the corpora allata, which is the gland that produces juvenile hormone, which is crucial for many things in insects, but for purposes of our study, it was really important to test for ovary development in infected versus uninfected hosts. And what she found was that the corpora allata in foundresses, so these are the females that are initiating nests in the spring, as expected, they should have large glands because they're producing juvenile hormone for reproduction purposes. The giants, which are the future reproductives that are going to overwinter and then become queens, the next one, were slightly less developed in workers who usually do not have their own reproductive needs, or they at least don't try to directly have fitness, it was slightly lower. And then for workers infected with one female parasite or one male parasite, we had some outliers, but we see a trend to a slightly smaller corporalata, and it did not have any effects on males or males that were infected at all. So we had a little bit more hint of where to look for mechanisms. So Rafael, which was a visiting scholar in the lab, who's now doing a postdoc in France, and Hannah Cook, our technician in collaboration with Yasser Ahmed from Syracuse University, we set out to look for gene expression between male and female hosts if they were infected by parasites. So we collected giants, these future queens, infected workers and uninfected workers, and we also collected infected and uninfected males. We were able to dissect out their brain capsules. This included the head capsule without muscles. We cleaned them out. We extracted the RNA, and then we sequenced and analyzed the transcriptomic data. Here on the right, you can see a male wasp. It has these curly antennas, which are typical of males. The coloration is different. And you could see, if you squint a little, you could see a parasite sticking out of its abdomen here. And here you see the female parasite where the female host, where the parasite is a little bit more noticeable. We were able to sequence 24 females and 16 male hosts in these different categories. Divided into these categories, we had very high coverage, and it aligned very well to the host wasp genome. So we were very encouraged by this. And here are the first results that we have. So we found that our dimension plot over here teases out male and female wasps and gene expression, which is what we would expect, of course. But the second dimension is really grouping giants, so the future queens, which are here in orange, with the infected workers, and then the uninfected workers are here in green in a separate group. So based on Amy's work, they had found that the infected workers are in between giants and workers, so they're a weird manipulation caste-related phenotype. But what we found is that they're really more alike to the giants, except for this castration part, which I'll address briefly later. So this was pretty exciting. It seems that infected wasps their plasticity is being manipulated to turn them into these weird giant-like individuals in some ways. And then on the right side, we see uninfected and infected males, and overall they're grouping, they have some grouping on their own over there. So we were very encouraged by this. And the next thing we wanted to look is for whole gene expression according to host sex and infection. I know that this graph is a lot to look at, but what I'd like to point out over here is that we see two groups here to begin with. And this first group over here is males, and it divides up into two subgroups, infected males and uninfected males. And the other group is separating into giants, which are these green individuals over here, this group over here, and then it's grouping infected and uninfected workers separately too. So this also is giving us confirmation for these transcriptomic profiles, which are pretty distinct in the 5 categories that we're looking at. So it's dividing as expected per sex, males and females, but it's also dividing up into infected and uninfected individuals with distinct transcriptomic profiles. So we're very excited. So then we wanted to test for pairwise comparisons that would give us hints about which genes and associated to which functions would be manipulated by the parasites to obtain and manipulate the phenotypic plasticity of the host to make this intermediate caste that should overwinter, should live for a long time, to be able to infect more hosts the following spring, but not do any of its usual social tasks and be castrated. So what we first compared were worker and giants, the usual two castes that we have in this system. And we found differences in lipids, in sugar metabolism, and in caste-associated genes, which is what it's expected, short-lived workers that don't overwinter compared to long-lived future reproductives that should overwinter, and they need lipid development to be able to overcome the winter. So this was very exciting, of course. Then we compared infected workers and uninfected workers, and we saw differential gene expression. And what was really compelling here is that we started to get signatures for the immune system, which is what is expected, and for neuromodulation too. And then when we compared infected workers and giants, if you remember from the dimension plot that I showed, these two groups were very close to each other. The differences here were in immune system and also in reproduction, because we know that giants will develop their ovaries the following spring, while the infected workers are being parasitized. So we were starting to get the first signatures of where we should look at. Then we also compared between male and infected males and uninfected males, which we really had no idea what was going on. And we know that they don't affect the behavior of a male host, they don't affect the physiology, but they do develop inside the host, although they can't extend the lifespan of its host. So it's kind of a dead end if you're a female parasite to parasitize a male host. That's short-lived. And what we found that the big differences between infected males versus uninfected males had to do with stress resistance. And this was one of the clues that will become important later, which is dopamine metabolism. And then when we compared males that were infected with females that were infected, we found that there is a difference in the D2 receptor, which is part of the dopamine system, which in Drosophila and in C. elegans has been associated to increasing longevity, to production of lipids, and to changes in reproductive effects. So we started to get a hint at which pathways we might need to explore for longevity, for example. The other unexpected finding we found when we started out with this system is that we started to see phenotypic effects of parasitic neuromodulation. So to the right, I want you to look at the abdomen here of this wasp. This wasp is infected and it has a tan colored abdomen, which is not something we usually see in this species. This is the North American species that's native. And to the left, we see an uninfected individual with its usual dark brown coloration. And to us, this was very exciting because there are papers of natural mutants in Drosophila in the wild, in which if they are depleted of dopamine, downstream, this affects the melanism pathway, and it creates this tan phenotype in Drosophila, which is what we were starting to observe here, but we needed to test for this. And these tan mutants in Drosophila also have an increased lifespan that has been associated to less circulating dopamine. So we were very excited to test for this. So the first thing we did is we wanted to quantify these changes. Well, it may look tan, but we really wanted to, as good scientists, to quantify if this was something that was happening. So here what I have is the first, in dark brown, we have control workers, and in tan, we have parasitized workers. We first compare an abdominal segment here, a part of the abdomen that usually doesn't change color. It's a red coloration. It's very noticeable and it's very consistent for the species, and we really didn't find much changes in the first figure for this part of the abdomen between infected and uninfected hosts. But when we looked at these tan regions, we did find significant changes where using a spectrophotometer, both for color and for brightness, we see differences where indeed infected wasps have a tan coloration. So that was very exciting. So then we dissected out brains of infected, uninfected workers and giants, the future queens, and we wanted to quantify biogenic amine levels to see if there was instances or some hint of manipulation by the parasite. And this was in collaboration by our team with Chris Jernigan and Colin Brent. And what we found here is that there were no changes in octopamine, which is the first figure, between control workers. The tan ones will always be the infected ones and the giants. And if you look at the second top figure here that I'm highlighting in dopamine, we saw that both infected workers and giants had higher levels of dopamine than the infected workers. And to us, this first was confusing because I just told you that the infected wasps should follow what they found in the Drosophila tan mutants, that they have less circulating dopamine. So at first we were confused by this result. And we found some instances of differences in tyramine and no differences in serotonin. But what we couldn't figure out at first was why there would be more dopamine in infected individuals that show this tan phenotype. Well, it turns out that when we look at gene expression of aromatic acid, amino acid decarboxylase, this gene that is associated to the dopamine and serotonin pathway, and to tyrosine decarboxylase, it turns out that there is a lot less expression of these genes that are what mediates these catalytic steps in the dopamine pathway in these infected workers, which are tans, and the future queens, which are in this light cream color. And then when we started to look at other important vesicles, transporters of dopamine traffic, so for example, dopamine DAT, which is this dopamine transporter, it turns out that if you can look at my cursor here, infected workers have less gene expression than even the future queens, and even a lot less than the usual control workers that are not infected. So it seems that even though the parasite infected wasps may have more dopamine, the dopamine seems to be trapped, it doesn't circulate. And it seems that part of the parasitic manipulation is to limit this biogenic amine traffic, which would have strong effects in neuromodulation. And it allows us, it provides some of the first results that match this depletion in dopamine in the downstream in the melanism pathway, which less dopamine would result in these tan phenotypes too. So very exciting, but we really wanted to look at how this would tap into actual neuromodulation. So the last part I'm going to be talking about is, these are brand new results and we're just getting going, but which gene networks would be able to be associated to manipulating a host phenotypic plasticity to make it not be social, to castrate it so all the energy can be allocated to the fitness and survival of the parasite, and to overwintering so it becomes a vessel to infect other social wasp hosts the following spring. So we wanted to target our key plasticity targets were, well, we wanted to look at gene networks that would be associated to longevity and to energy allocation. We wanted to look at genes that could be associated to overwintering, that usually giants would use to be able to survive the winter successfully, to neuromodulation, and of course to the immune system. So Rafael led this effort. And here we have a heat map of module trait relationships. I know that there's a lot of boxes here, but maybe what is important, the take home message, is that we have our 5 distinct categories, each one here in a column. We have our workers, our infected workers, the giants, which are the future queens, the males and infected males. And here we could see patterns of gene expression. So we started to look at some of these. So if we look at, for example, the first module, it has genes that have been associated in the literature in insects and in other systems to lipid storage and to caste determination. And here, what we could see is that it shows differential gene expression in infected workers, and it has slightly less in giants, but it has less gene expression or less activity in these genes in the usual workers that are not infected and are short-lived. We were also, and following this logic where we could look at differential expression of these modules, we were able to nail down some key candidate networks for parasite development, for immune system, which would explain some of these encapsulation responses, or for the parasite to be able to evade the immune system of the host and remain undetected while developing inside the host. We also found that this one, we're very excited about this purple one, which it has a lot of genes, but it shows neuromodulation in our biogenic amines, which of course dopamine is of strong interest. We also have indices for energy allocation and parasitemia, that is infection, circulating hemolymph, and then of course stress responses and hormonal metabolism. So we're getting close to getting some answers. And then when we look at this heat map, and at the same time we look at this module clustering, so of these modules, which ones tend to have, may have links and relationships between these gene networks, I wanted to point out these two here on the top, which are highlighted by these red squares. These are modules for hormonal metabolism and energy allocation, which we would expect would be similar in infected workers and infected males, because they have to do with the parasite developing and metabolism. And indeed, we see similarities in these two categories. Here are the infected males and the infected workers. But we also know that infected workers live for a long time, and they are castrated and they change their behavior, while the infected males don't have any of these traits. They live for a very short time. Their behavior and physiology is not affected. So then we looked at modules for lipid storage, for caste determination, and then for neuromodulation. And this group over here that I'm highlighting are gene networks that are closely related, or at least more related to each other, may have some links. And here, if you look at gene expression, which is indicated by the color coding, it's different in infected workers compared to infected males. And lipid storage is important for overwintering and for longevity. Neuromodulation is important for changes in behavior to hijack the phenotypic plasticity of the host. Biogenic amines and these transporters and vesicles that will transport biogenic amines, and the immune system. So it seems that we're getting very close to getting to the answer of which how the parasite is really hijacking the phenotypic plasticity of the host. So I don't want to give you a headache with this, but we're starting to really look into these gene networks. This is the gene network underlying neuromodulation, where we can start to connect the dots and hopefully find some master regulatory genes, hopefully. This is the one for caste determination. So again, I don't want to give you a headache again, but just to give you an idea that we're getting close to examining specific relationships among genes within these networks, which are important for caste determination and neuromodulation, which are the key elements that the parasite needs to manipulate. And so far, what answers do we have to our questions? Well, the first thing is that we know that gravid females can hijack the sensory system of their host that should be used for social communication, nest mate recognition, and instead use them so the infected host can localize nests of conspecifics to transfer the offspring of the parasite. We know that the parasite larvae don't seem to discriminate. So whenever they're dropped off, that's what they try to infect. They can infect the invasive hosts, but there seems to be some differences with the native host. And then according to mechanisms, we know that infected workers have a giant-like brain. So we saw these neural gene expression, these transcriptomic profiles that I show you, they seem to be more gene-like, not an in-between as much, but more towards like a weird gene-like phenotype. And then it seems that the hijacking of these different modules, gene networks, has to be what puts together and secures the success of the fitness of the parasite using these, the phenotypic plasticity of its host, in this case, causing a negative effect on the host, of course. So what do we know so far? The female hosts have an extended lifespan. The male hosts don't. They cannot be manipulated to extend their lifespan. Female hosts have behavioral changes. The male hosts don't have behavioral changes associated to manipulation. There's mild neuroanatomical changes in the females, but strong neuroanatomical changes in the males. And then the gene networks that seem to be crucial for manipulation of the females, which is the ideal host, are neuromodulation, longevity, caste determination, reproduction, and immune system, while the ones on males that the parasite seems to be able to manipulate is energy allocation and overcoming the immune system of the host. So just before I finish, what are the next steps? Right now, we want to spatially localize where neuromodulation is happening in the brain through a combination of immunohistochemistry and single cell RNA sequencing, which can give us a better spatial idea of how this is happening in the brain. We also want to functionally test when we have a little bit more confirmation of candidate genes that the parasite would be manipulating. And excitingly, we have a draft genome of the native parasite, which is led by Maria and Emiliano. So we also have the parasite's gene transcriptomic profiles that we can also compare. So as the parasite develops, we can compare its gene expression and also the gene expression of the host to see how this is in tandem working out. So very exciting. And with that, I would like to thank, it really takes a village to do this type of work. Again, this is something we just started now in 2020, and I'm very grateful for our wonderful group. And before we start with questions, I would just like to mention that I am very committed to diversity in STEM, and I would be very happy to talk to if there's any students that are interested about talking about experiences, Latin American students that want to pursue a PhD program or such graduate opportunities in the United States. I would be very happy to talk to anyone that I can be of service to. So thank you for listening, and I'd be happy to take any questions.
Thank you very much, Floria, for such a great talk. If you unsure, it'll be easier for, here we can see everyone. Well, now it's time for questions. And so just a reminder, please tap the question mark in the chat, and I will call your name. And so you can ask your question. Please state your name and affiliation before asking your question. We're trying to build a community, so it'll be great to get to know each other. People watching us on YouTube, please type in your question and we'll get it relayed by Carsten, helping us overseeing the YouTube channel. Okay, so is there any question in the room? I'm gonna open Carsten.
Yes, thank you very much for a great talk. So, such a nice series of experiments and studies. My name is Carsten Schradin. I'm a researcher at the CNRS in Strasbourg, France, studying mammalian social evolution, mainly doing fieldwork in South Africa and comparative studies. In the end and during your talk, you indicated that it seems that the parasite, the one thing it is doing is to change energy allocation. And this is then probably the reason why the infected females can survive so much longer. And this indicates, even though you did not measure it directly, that the the workers themselves, they spend all the energy to produce as many future queens as possible, and thus they don't have enough energy to survive through the winter. Is that correct? Do you have any information on this? So the reason why, so the evolutionary question is, why do workers die, not survive? From the functional point of view, is simply they invest all their energy in the reproduction while they can help, and in the help of reproduction of the colony.
Yes, so I think this, the answer to this is a combination of things. The first thing is that the life cycle of parasite is early in the year, so spring and early summer, and the available hosts are workers, which are usually the ones that shouldn't overwinter and shouldn't survive. So they have to find a way to make short-lived individuals tap into the phenotypic plasticity of the host and instead transform it into a giant-like phenotype that should be able to survive. There are instances, and castrate them, as you say, because that way they're not using, reproduction is so costly that they can allocate the resources for the parasite's fitness. Sometimes there's parasites late in the fall and they can infect future queens. So they technically shouldn't have to manipulate their longevity, but they would still need to castrate them because otherwise they're going to lose all that energy. So I don't know if that gets to your question per se.
Yeah, I mean, your study system is a very nice example against the pathogen control hypothesis that has been proposed in Trends in Ecology of Evolution a few months ago, which is a nice hypothesis, but doesn't explain everything. That says individuals should die early to avoid the transmission of pathogens. In this case, it's exactly the opposite. The pathogen manipulates the host to live much, much, much longer. Do you know, are there many other examples in the literature on pathogens that manipulate the host by spending energy instead of reproduction? And in this case, I also see helping as a kind of indirect reproduction into survival. Are there many other examples? Or is that very unique?
I can think of several examples, at least in social insects, where parasites do manipulate castration. So they do castrate their host. So if we can think of what are metabolically very energetic necessities of an individual, it's usually the brain's pretty expensive metabolically, and reproduction is too. So it seems that parasites have found ways to reallocate energy, and one way to do that is to castrate. And in some other parasites, we have also seen changes in the brain too, where the individuals have smaller brains. We saw that in our males. So perhaps, if, to answer your question, male hosts don't live long, they're not being manipulated, and they don't really have ovaries that could be castrated. So the parasite seems to be manipulating the brain. They have very small brains compared to uninfected males. So this also seems to be another way of allocating energy from very expensive systems into what the parasite can use. So I know that at least castration is something that is used by parasites, at least in some invertebrate systems, to reallocate energy.
Yeah, and is this energy then used to increase longevity or to produce more eggs of the parasite, or both?
We don't know. Yet. I couldn't give you a definitive answer. We would expect that some of this energy would be reallocated, but if we think about this giant parasite developing inside, sometimes it takes almost the entire abdominal cavity. When we do dissection, it's hard to understand how that works out. They seem to be tapped into growing around the digestive system of the host. So we think they feed off from hemolymph, but we're also not sure if they're also getting nutrition from the host digestive system. So we're at that stage where we're trying to explore, but at least for the growth and the development of the parasite, they do need to consume a lot of the resources of the host. How that would go into longevity, I think when we get better hints of how the neuromodulation is happening, and also like you and I were talking about earlier before the talk, there should be some blood-brain barrier situation that we're trying to figure out with circulation and hemolymph, that will give us a better idea of how that would be happening.
Okay, thank you.
Thank you, Carsten. Our next question comes from John Strassman.
Hi, Floria. That was a great talk. It was so much fun. I'm Joan Strassman. I'm at Washington University in St. Louis. I used to work on wasps, but haven't for about 20 years, work on social amoebas now. So it's just so exciting to see how the field has progressed and how much you've done and been able to achieve. I mean, it's just so exciting. So my question is, I saw evidence of strepsipterans a lot in Polistes, but I don't think I ever saw them in the neotropical swarm founding wasps. Do you have any sense for, maybe you said this, but if you could elaborate on how widespread strepsipterans are in this social Vespidae?
Yes, absolutely. Hi, Joan. It's such a pleasure to see you. For those of us that are WASP people, I kind of grew up reading Joan's work and David, so they've been a true inspiration to my work always, even though we forgive you for not being at WASPs anymore. We love your social amoeba work. So interestingly, and this is one of the things that has been one of my pressing questions, so I'm glad you brought it up. There are neotropical strepsiptera that attack neotropical species. We have seen them in these single founder small groups, and in the swarm founders, they're very common. They've seen them in Agelaya, Parapolibia, all these different species. But what's really, so they seem to be widespread, and they also infect solitary wasps and solitary bees too. Of course, different species, they seem to be more specific. But the interesting thing here is, if the neotropical hosts don't experience winter and they're asynchronous, what is the manipulation that they have to do for the neotropics when they don't have to overcome overwintering and the temperature and the lipid depletion and all these other things? So in the future, you kind of read my mind, because we want to test what the neotropical host, what is the manipulation of those compared to the ones that experience seasonality. So it's a lot more widespread. And now that different WASP researchers are more aware in the tropics of this, they're finding them more. So I think there'll be strong collaborations in the future.
Do they show up on the bodies the way the ones on Polistes do?
Yes, they do. They do. And when I show them to, for example, WASP researchers and the social insect meetings, they're like, I've seen that in such and such species, and it's showing up a lot more than what we thought.
Are they more in the bigger ones? Because like Polybia is really small.
They're even in Miscasiderus, which is a tiny, tiny wasp. So I think the size of the parasite, the species changes, but they can affect single foundress or small groups versus swarm to solitary. They seem to be doing well in different hymenopteran hosts. Yes.
Thank you, Joan. Our next question comes from David Feller.
I'm just moving over to Joan's computer here so we don't have to mess with the sound controls. Yeah, I'm David Queller. I'm also at Washington University in St. Louis. Also used to work on social wasps, but now currently work on social amoebae. So hi, Floria. That's a great talk. I remain a little bit confused about the similarities among gynes, workers, uninfected workers, and infected workers. So you had this nice brain transcriptomics, multi-dimensional scaling plot, which put the infected workers very close to the gynes. And so I have two questions about that. The first one is, I'd like to understand better why that is, or what that reflects, given that you've got sterilized workers and they're more like, reproductive gynes, which seems very weird. And the second question is, how robust is that? Because I remember the next slide you went to, and I think it was the same data. Was it the same transcriptomic data? You had hierarchical clustering of the different types. And in that case, the workers were more, the uninfected and infected workers were more similar to each other than they were to the giants. So what's the difference between those two things?
Yes, that's a really good question. So I think that our data is a combination of things. First, the first plot was it was the entire transcriptomic profile, meaning the ones that had differential gene expression and the ones that didn't. That hierarchical one, it shows that, and some of our other results only show differential gene expression. So maybe that's good that you bring that up, because I realized I didn't point this out. So we have some tests where there's only differential gene expression, so differences between the categories, and some that look at the entire transcriptomic profile. And that's our hierarchical clustering that you saw. So based on that, infected and uninfected workers are clustering more next to each other because they're both indeed workers, right? They are, we do know that in social insects, nutrition, right? What they get fed as larvae and originally being destined to be workers should have an impact on their transcriptomic profile. But what the parasite is doing is trying to manipulate certain parts. So what would be the giant-like parts here? It would be the longevity aspect, the lipid development, and finding ways for it to be able to overwinter and overcome the stress. But the difference is that they need to be castrated. They can't be like a giant. We do know that giants have temporary, I wouldn't call it repression, but at least inhibition of reproduction when they haven't developed their ovaries before they become foundresses the next spring. So I do agree with you that it might be a little bit more confusing. So for differential gene expression, they seem to be more like giants, except for the reproductive parts, the reproductive physiology part. But when we compare the whole profile, meaning differential expression and the genes that are not different from each other, then they group with workers because indeed they are workers. So I think that looking at specific modules will give us the answers, because they're kind of Frankenstein-like, right? Where you're grabbing the traits that you need from a trajectory for a giant and maybe retaining some of the things that work as a worker and not manipulating them. And that's probably why there's this weird in between. I don't know if that gets to you.
Yeah, I think it does. Although I'm a little bit puzzled that in theory, genes which are not differentially expressed, they're expressed exactly the same, should not affect the classification either in the MDS or the hierarchical clustering. So it must be that some of these genes are differentially expressed. They don't have exactly the same values, but they're not significantly different. And that's the difference between your two data sets. Probably.
Yeah. Mm-hmm. Yeah, no, you're right about that. Maybe I'm simplifying it to black and white, and what we're seeing is these shades of gray that, depending how we analyze things, seem to be. So perhaps it is an in-between. Most of our analyses show more towards the giant-like phenotype, except for the reproduction. So we see the lipid profiles, we see the longevity part, we see the energy saving kind of metabolism. We found some signatures with sugar metabolism, which switches to, what is the two glucose molecules called together? So there's more trehalose rather than glucose being used. Workers use more glucose. We see more trehalose transporters in the giants and in the infected workers, which hopefully helps their metabolic issues, right, to be able to overwinter and such. So I agree with you. Maybe it is an in-between, but it's catching a lot of the giant-like traits that it needs to survive.
Yeah, thank you.
Thank you, David. Our next question comes from Sulema.
Yeah, hi. Sulema Tang Martinez, retired from the University of Missouri in St. Louis. And I have, as usual, unfortunately, several questions. And so I'll ask them one at a time. And Floria, fascinating talk. Thank you so much for doing this. Really, really interesting. My first question is, given that a given wasp can be parasitized by more than one parasite larvae, I'm wondering if there is any competition between the parasites when there's more than one in a given wasp. And if you look at a female parasite, when she parasitizes the host, do they lay only one egg, or can the same female lay more than one egg? And that's all related in terms of, obviously, in terms of competition among the parasites.
Yes, so actually, this was something that I originally had in the talk, and I took it out because I would have you here for 2 hours, and nobody needs to talk about parasites for 2 hours in their lives. But there is a very strong difference. First of all, there are super parasitized individuals, and I think this has to do with the availability of hosts. So we do see them in nature. We kept them only to female parasites and to one at a time, so we would have comparable neurogenomic profiles, because otherwise it could get very messy. But you're right, there's super parasitism. We have found 9 in an abdomen. I don't even know how anything fits in there. It's insane. I think all my undergrads were traumatized the day when we found that. And it's a combination of male and female parasites. But here's where it gets really tricky, and something that we want to look in the future. The needs of a female parasite: you need your host, you need to overwinter, you're an obligate. And we've seen even females with three parasites that are females. We've caught them the following spring when they're en route to infecting. We've seen them visiting nests, and we have found them. So super parasitism is possible. There could be competition, which is normal in parasitoids too, but somehow we've seen them function if they have several parasites of the same. We have also seen in the late fall, some of the pictures I showed had the whole from a male parasite that emerged, and female parasites remain. Those could die because of infection when the hole is open. Sometimes they get in, pathogens and other things, so there could be more conflict. But the real conflict I want to point out is that you're a male parasite. All you need is, you're like an alien growing inside the host, and you need someone to feed off, and then you pop out. You don't care what happens to that host. So those hosts that are infected by males, they die also. They don't overwinter because there is no manipulation by the male host. It just needs an eating vessel, pretty much. So if you are a female host, a female parasite, you do very well inside a female host. But if you're a female parasite and you infect a male host, you're in trouble, because the male host will die and you won't get to live to pass on, your genes the next generation, the following spring. So that's a dead end. It's even, I don't want to say maladaptive, but it's the wrong choice, right? If you're a female parasite inside a male host. If you are a male parasite, you could do okay if you're inside a male host, because then you emerge too. It might be late in the season, there's less chances for mating, but you could do that. But the ideal host ends up usually being the female. And we don't know if the infected wasp with male parasites die because of an infection, because they leave these gaping holes, or it's that the longevity manipulation isn't happening. And we would like to tease those apart, because we don't have an answer yet.
Yeah. So my next question is whether there's any evidence that when these parasitized wasps are coming into nests of unparasitized wasps, whether there's any evidence that the unparasitized wasp can recognize that the ones coming in are parasitized and reject them, attack them, or kick them out of the nest, whatever.
That is a wonderful question, because we know, and even Dave and Joan have worked in some of these initial dominance hierarchies where there's usually one dominant foundress and the other ones become subordinate until they produce workers. So in these early groups, sometimes there's usurpers or there's joiners, depending on the wasp system and the species and all these other things. So there could be encounters where individuals need to recognize this one's from my group or it's an intruder, and we don't know what the intruder wants. That does happen, and they have very sophisticated hydrocarbon profiles, odor, where they can recognize. So even if a wasp wasn't infected, if it doesn't belong to that nest, it should be attacked to begin with. But here is where I keep getting convinced that these parasites are crazy. You know that they usually visit unattended nests when the foundresses are away most of the time. That's how we catch them, because we see these weird wasps. It's like visiting, and they have this funny flying pattern where they visit unattended nests, and sometimes they stop and we're like, that one's infected, because they usually just fly into their nest and they start to do things, but the behavior is different. So what we have seen in the field, and this is how we catch them, is these weird wasps that kind of like hop between nests flying around. So I think that I do agree with you. If a resident wasp on its own nest would be there, they would be attacked, because they don't belong to that nest to begin with. That and the fine tuning of recognition if they're infected, it could be, but we would have to figure out what type of experiment to run to tease apart conspecific intruder and conspecific intruder that's infected. And we would have to figure that part out. But they do recognize that they don't belong in that nest.
Yeah.
We know that.
Yeah, I also was wondering if in part of what I was thinking with that question is whether being parasitized actually causes the host to produce additional odors that are not necessarily their own odors by which they could be recognized as foreign, but that they actually have a different odor as a function of being parasitized, which I guess is possible also.
And it is actually, there's some wonderful work done by Brazilian researchers, which are the other amazing wasp researchers that there are. And they found hydrocarbon profiles that are very different between infected and uninfected. Yeah. What we suspect, and we would like to test, is that this hydrocarbon profile may be more giant-like too. So okay. Individuals that are infected usually stay in their natal nest before they go out the following spring and try to infect. And they don't get punished for not working.
Yeah.
And we think they get fed more. Again, this is speculation, but we think part of the parasitic manipulation is to trick them to think that maybe they're feeding future reproductives.
Yeah.
Maybe the hydrocarbon profile is also being manipulated to be more giant-like. So I'm a future reproductive, differential nutrition, feed me more kind of thing. And that also would explain why they don't get punished, because workers that don't work, they usually get policed and they're like, okay, get back. But these unaffected ones are mooches, and they get fed early on in their natal nest until they leave. Wow. So I agree that there should be a hydrocarbon profile manipulation that we want to test.
Yeah, that's really interesting. And then the last question, which I think you sort of maybe have answered, is the females overwinter, live longer, and the female parasite stays inside them, and mating takes place with the parasite still inside the female. Does the parasite's life therefore equal the wasp's lifespan, or is there any way that as the wasp is dying, they can leave and go elsewhere? Like, are they committed to, the wasp that they're in And once that wasp dies, it's all over for them.
Completely in sync.
For the
When-
females, especially.
Yeah. And once in a while, we've seen a wasp that's looking bad and it's infected. We're like, oh no, the parasites! We need to milk them so we can, because we know that once the host is dead, the parasite, the female parasite, will die very shortly after. Yeah, complete obligate endoparasite. It doesn't even have legs. It retains its larval form, completely dependent on the host. Completely, and we're seeing differences in the genomes of male and female parasites, which probably has to do with this crazy sexual dimorphism, the free-flying males versus the blob females that remain, you know, inside the host. Yeah, so they're strongly linked. Yeah. So they really need that vessel to keep it alive.
Yeah,
right?
no kidding. Yeah. Okay, thank you.
You're very welcome. Thank you.
Thank you, Zuleima. Well, next question was from Karsten, but his internet is not properly working. So we'll give him a minute. Maybe he'll be able to come back. Meanwhile, I will ask my questions. Thank you, Floria, first for your awesome talk. I have two questions. The first one, and you sort of talked about it before, so I would assume we can say that there is assortative preference for female parasites for a female host and male parasites for a male host, right? But my question is, how do they recognize a parasite? This is a female, and I need to just parasitize this female. So is there any, like, is it odor, or how does the parasite knows which one is the sex they need to parasitize?
Well, I think I may have confused you. Usually the target host is a female, no matter what. But if you're a male parasite inside a male host, you still have a chance of surviving. You're okay. But if you're a female parasite inside a male host, you're doomed because your host will die shortly. So I think this has to do with the life cycle. So very early in the colony, the first brood are workers and they're all females. And that's usually what the parasite affects. So I don't think there's much of a choice usually, because during most of the colony cycle, they'll produce several batches of workers, and then towards the end of the cycle, they'll produce the future reproductive females and the males. And then the males will go off on their own and they'll try to mate with females from other colonies. So there could be, and there are Italian collaborators, they have proposed maybe there's a bivoltine cycle of infection where there's an early cycle where they only parasitize workers, and then later on, there might be a second batch where they parasitize future reproductives. And in that case, there's an equal chance of a female or male larva host. And we don't know really, I mean, we gave them the wrong species and they hopped on no matter what. So maybe there isn't strong recognition by the larvae. It's just an assumption that if they get somewhere and there's a host, they'll infect it. And the usual chances is they'll get to a female worker host, which is what they usually need. Either as a disposable eating machine if you're a male parasite, or to manipulate it to be able to overwinter inside the host. It's like a giant coat, feeding coat that they use, right? Yeah.
But that's indeed very interesting, because then they really need to synchronize the life cycle of the host in order to get the best opportunity as in parasitize only females. I mean, there should be some sort of synchronization mechanism somehow that triggers them to start parasitizing.
So maybe I didn't explain this part well because of time constraints, but when after winter happens and the spring comes up, who wakes up? Usual reproductives, right? The giants, they'll become foundresses, they'll start their own nests, and then infected wasps wake up too. So everybody's waking up at the same time, and the infected ones wait until the uninfected ones start to lay their eggs and they have larvae, and then they infect. So it is kind of synchronized early on in the cycle. If it happens, and maybe this answers the question of why the male host is secondary, it seems to be maybe more of an error where it's late in the season and there's potential male larvae. So that does happen, but it's less frequent than early on in the cycle to secure you have the right host to be able to survive. And do
Awesome.
Yeah.
That's pretty interesting. My second question is a little bit off this part of the topic. At the very beginning, you mentioned cognition, but I might have missed it in the talk. I don't know. But I was thinking, okay, if there is a change in brain size, is it affecting anything in the cognition, or what is the parasite affecting in relation to the cognitive abilities of the wasp?
Yes, originally we thought that these females that are not being social, they're not being exposed to their usual social interactions, right? As soon as they emerge as adults, they kind of stick around, but they don't interact, and then eventually they leave their natal nest. We thought, well, there's social isolation studies, not only in wasps, but in a lot of other organisms, especially with what we're dealing now, where brain regions get diminished, right? Their areas are smaller if they don't have social interactions. So we thought, well, if we compare usual workers, they come and go with their nest, they antenate with their nest mates and everything, compared to the ones that are not really interacting as much, we might see differences in the olfaction related brain regions and the visual ones, because they're not going out to forage, and we didn't find them, which was surprising to us. But at the same time, they're staying in their natal nest, so they still have the olfactory cues, and they do see individuals come and go. So that wasn't as drastic as we expected. But in males, since our hypothesis is that since they can't tap into castrating their host, the other energy they can allocate is more brains. And that's why male hosts have really small brains, and some of the sensory regions are really small. But it could be a side product of the wrong host, and they're just grabbing energy from when they can. And we know that the brain's really energetically expensive, and it doesn't matter if the host has small brains if you're a male that is short-lived. So that's what we're thinking. They don't have the right thing, so they tap onto the next best thing, which would be taking energy that should be allocated to developing the brain.
Interesting. Thank you very much, Floria. Is there any other questions that we have in the room? If not, I will turn off the YouTube live, and then we can just chat informally. I don't see any question mark coming into. So Floria, thank you very much for an amazing talk. I think this is an incredible system with tons of opportunities. I thank you really for accepting the invitation to participate in the FIND seminar, and we hope to see you here as a participant as well. I will turn off the YouTube live and